All right, so as we move into the fourth part of this lecture, we're continuing to work through the initiation process of translation. So, up to this point, just to remind you where we're at, we've got the first step that took place was where the transfer RNA was actually charged with its specific amino acid. So in this step, the amino acid is going to first react with the adenine triphosphate, and that is going to produce an aminosyl adenine monophosphate plus two additional phosphate groups that were cleaved off. Then what's going to happen is in the second step, the amino acid is going to be transferred to the transfer RNA, which is this green kind of Ghostbusters looking type configuration. And that adenine monophosphate that was created is going to be released. So this is where we start to see our energy transfer that really characterizes the transfer RNA charging process. So at the end of this process, the transfer RNA is going to be linked to its appropriate amino acid. That led us into initiation, and initiation is where all of the components were going to come together and assemble on the ribosome. So the ribosome is made up of two subunits. One is a large one is a small one. And then the components that are going to come together include messenger RNA, the initiating transfer RNA, guanine triphosphate, initiation factors 1, 2, and 3, and then the small subunit and the large subunit. So up until this point, we've had the initiation factor 3 is going to come together and bind to the small subunit, and that's going to prevent the large subunit from binding at that point. Then that interaction with initiation factor 3 in the small subunit is then going to allow that small subunit to then attach to the messenger RNA strand. And finally, the transfer RNA is then going to move in and going to form a complex with initiation factor 2 as well as guanine triphosphate, which is another energy source. That complex will bind to the initiation codon, and at the same time, initiation factor 1 is going to join the small subunit. Then, as we start to wrap up this initiation process, after these complexes are now in place, all of the initiation factors 1, 2, and 3 are going to disassociate from the subunit complex. GTP is going to be hydrolyzed to GDP, guanine diphosphate, and the large subunit is then going to come in and join the initiating complex. So that really is a good summary of what's going on with the initiation process. There's lots of moving parts, but once you start to break them down and get the fact that this one comes in before this one, this process really is actually quite simple to memorize. So one thing to keep in mind as you're struggling to grasp this initiation concept is that this is a highly regulated process. The more regulation factors, whether it's an initiation protein or a subunit or the actual mRNA strand, all of those things have to come together in the specific order for translation to take place. If some part of this regulation process doesn't take place in the correct order, that's where we start to see genetic diseases take place. All right, so just keep in mind at the end of step 2, which is initiation, the ribosome is fully assembled and is on the mRNA strand and is also attached to the first transfer RNA that is going to bring in that initiating methionine. So as we kind of back up and just talk about the Shine-Dalgarno consensus sequence a little more in depth, this consensus sequence occurs upstream of the initiating methionine. And the sequence is going to be very different depending on what species we're talking about and what gene within the species genome we're talking about. So as we are trying to synthesize and fix translation processes, it's important to keep in mind the Shine-Dalgarno consensus sequence because this is going to be required for attachment of the small ribosomal subunit. So we can see that Arabidopsis in the B gene has a much different sequence than the lactic acid gene in E. coli. And so that consensus sequence has to be very specific for translation to take place. All right, so one other quick note before we proceed into elongation is as we look, take a step back out and look at translation as an overview, on our mRNA strand, which is oriented 5' to 3', we've got our start codon, which is the initiating methionine, and then our coding sequence, which is going to produce a polypeptide strand, and then we have our stop codon. And after that stop codon is going to be a 3' untranslated region that's going to contain a poly A tail at the end, which is a string of adenines that is going to interact to enhance translation. So as the translation process is taking place and we've got assembly of our ribosomes and cap binding proteins, if we're in eukaryotic systems, those are further proteins that are going to help regulate the process. The actual messenger RNA strand that has this poly A tail has the ability to wrap back around, bind to the top of a cap protein to help this whole process of translation take place. So it will actually enhance translation. It'll help speed it up or slow it down, depending on the need of the gene. So keep in mind that the ribosome is going to have 3 major sites that are occupied by transfer RNAs. So when we brought the small subunit and the big subunit together, and the mRNA strand is sitting in between those 2 subunits. Within that ribosome, there's 3 different spaces. The first space is going to be the aminoacyl or A site, and that is going to accept the incoming transfer RNA. So that's going to be on the right side of the ribosome. So it's a little bit different than if we're looking at it from a 5' to 3' region. So that A site is going to set on the 3' end because the ribosome is moving towards that 3' end. The second site within the ribosome is going to be the peptidyl site or the P site. And this is going to be where the peptide bond forms between the amino acid and on the A and P site. And finally, the E site is going to be on the 5' end of the messenger RNA because the ribosome is moving away from the 5' end. And this is going to be the E site or the exit site. And this is going to be the site that actually removes the uncharged transfer RNA after the amino acid has been bound to the previous amino acid. So the third major step is going to be elongation, and this is where the amino acids are joined to create the polypeptide chain. So the way that this happens is that ribosome is going to move from the 5' to 3' direction, and every time it moves to an A site, that new transfer RNA is going to come in, bring its amino acid, and it's going to bind to the polypeptide chain. So the elongation factors can actually help with this process. And as the ribosome is moving from codon to codon on the mRNA strand, we are creating peptide bonds between the amino acids that are residing in the peptidyl site or the P site, as well as the A site. And the enzyme that is doing that process is called peptidyl transferase, and that is going to be an enzyme catalyst. So it's actually going to be creating that peptide bond. And then once that bond has been created, translocation takes place. So the ribosome is going to move down the mRNA strand to the next codon position, and that is going to require the elongation factor G to be in place to help with that process. So here is the entire process of elongation. So if we start up here at the top, we've got our initiating methionine transfer RNA complex that is going to occupy the P site or the middle site of that ribosome. The initiation factor, but the guanine triphosphate is going to be present, as well as the charge transfer RNA. They're going to form a complex that enters the A site of the ribosome, or the site that is on the furthest right, or the 5' end of the ribosome. After the charge transfer RNA is placed into the A site, then guanine triphosphate, or GTP, is going to be cleaved to GDP, and the complex is then going to be released. After that, the complex is then going to regenerate and is then ready to combine with another charge transfer RNA. So as we start building those peptide bonds between the amino acids in the P and the A site, the transfer RNA in the P site is going to release its amino acid. Then the ribosome is going to move down the mRNA strand to the next codon, and the transfer RNA that was in the P site is now going to be in the E site, from which it moves into the cytoplasm. And finally, the transfer RNA that occupied the A site is now going to be in the P site, and the A site is now ready and open to receive another transfer RNA. So the conclusion of the elongation process is at the end of each cycle of elongation, the amino acid that was in the A site is going to be added to the polypeptide chain, and the A site is then free to accept another transfer RNA. So this process will continue as long as there is a coding region of messenger RNA.